Mission Bio's single-cell genome editing analysis pipeline is a complete software solution for the detection of single-cell edits. It can help determine the rate of on and off-target events, as well as the rate of co-occurrence. The genome editing pipeline can generate 4 different reports: single-sample base editing, single-sample knockout, multi-sample base editing, and multi-sample knockout. This video will go through the 2 knockout reports, starting with the single-sample report. Note that the knockout reports only focus on indels and do not report out SNVs. Starting with the first page, or the summary page, we can get some general run details, including the number of cells that were called, the mean reads per cell per amplicon, panel uniformity, and the percent reads mapped back to target. The left column of this first page goes into more details about each of these run metrics and includes a full breakdown of each amplicon's performance. A help icon can be found at the top of each set of metrics. This link will take you to a support article giving more details about the metrics and how they can be interpreted. Taken together, these metrics can give you insights into the quality of the run itself, both on the input level and the sequencing level. On the right-hand side of this page, we begin to see some of our editing-specific information. This first table is a summary of editing, which details all edits, both on and off target, per target group, that was captured in the run, showing a breakdown of how it appeared through all alleles and cells. This table begins to provide information not available through bulk analysis, such as the number of cells edited, the zygosity status, and the co-occurrence of edits. This particular experiment had 3 intended targets, and each guide RNA also had 1 predicted off-target. The target groups include both the on and off target for the guide RNA. With this table, and with all tables in the reports, you can show and hide different columns using this drop-down menu at the top. You can sort the data in the table by each column, and you can download the data from various file outputs right from the HTML. Underneath this is a bar graph displaying the number and percentage of alleles that were edited versus unedited per target group. The target group can be shown and changed using the drop-down menu at the top of the plot. All plots are generated using Plotly, and Plotly-based tools can be found in the upper right corner of all plots to help visualize the data in different ways. Moving on to the second page, or the advanced page, we begin to get more information about the editing that occurred. The top 2 tables detail the top on and off target variants captured, showing information at both the allele and cell level for each variant, as well as the percentage of cells that were seen to be either heterozygous or homozygous. Beneath this, we can see a histogram of the indel lengths that were captured per target group, which again can be adjusted via the drop-down menu at the top of the plot. Directly below this is an on-target indel start position histogram, detailing where the indels captured began and about how many alleles had an indel start there. Moving down on the left-hand side is a top edit combinations per target group table. This table lists the most common co-occurring events per target group and shows the percentage of cells that had these co-occurrences. The last graph on the left-hand side displays the top 10 translocations in the run and the percentage of cells that these translocations were seen in. On the right-hand side of this page, we begin with a bar graph of the zygosity of all on-target variants captured. Below this is a more detailed view of the top 10 on-target variants captured and their respective zygosities. Next is a graph of the top 5 edit sites per target group and the percentage of cells these were captured in. Moving even further down, there is an on-target editing co-occurrence graph. This graph details the zygosity of the editing in each target group and the percentage of cells where each combination occurred. If you choose to process the data using our DNA plus protein assay, there will be an additional figure showing the protein expression versus target editing status at the bottom of this second page. The last and final tab of this report is the definitions page, which is present in every report as a reference tool. In it, every single term used and graph or table generated is defined and explained in more detail. When multiple runs have been processed with the same editing techniques, and you would like to compare the results, you can run the multi-sample pipeline. The knockout multi-sample report has 5 main graphics, the first of which compare the on-target editing status per target group between the 2 runs, and the target group can be changed using the drop-down menu at the top of the plot. The second graph compares the rates of the top 5 edits per target group between the runs. The third graph depicts an on-target editing co-occurrence graph. This graph details the zygosity of the editing in each target group and the percentage of cells where each combination occurred. One run is shown on either side of the central axis, and the runs can be changed using the drop-down menus on the side of the graph. Moving down is a top 5 edit combinations per group table. This table lists the most common co-occurring events per target group and shows the percentage of cells these co-occurrences are seen in per run. Lastly is a table of the top 5 translocations captured and the percentages of cells with each translocation per run compared. The second and last page of this report is the definitions tab, which again is here as a reference tool to explain everything in the report in more detail. Note that along with the report and the genome editing pipeline produces many output files. Importantly, there is a CSV file of the data contained in each table depicted, an HTML of each graphic, and an H5 file which contains information of all cells called in the run. A full list of the output files and what they contain can be found on our support site.